Good afternoon. Welcome to the NASA Social at NASA's Kennedy Space Center in Florida. We are here today to talk about the research going aboard the International Space Station, launching on SpaceX cargo flight here at 12:45 a.m. Eastern Monday morning. Not only is the International Space Station important for us learning how to do research, learning how humans adapt in low Earth orbit and in space for long-duration spaceflight, but it's also a great platform for researchers down here on Earth to do experiments. And so we have a list of speakers here today. They're going to be talking about their research flying Monday morning. And joining us here in the audience are our social media followers. We selected about 50 social media followers to come out here, get behind-the-scenes experience, to learn about what's happening here at Kennedy Space Center, and to learn about the cargo going up to the space station aboard SpaceX Dragon, and also, hopefully, see a launch at 12:45 a.m. Eastern Time. So, without further ado, I want to introduce our first speakers. We have Julie Robinson, who is the space station. She is the space station program scientist at NASA's Johnson Space Center, and Michael Roberts, who is the deputy chief scientist for CASIS. Michael, Julie.
It's really great to be with you guys here at Kennedy Space Center for the launch. And, we've got 930 kilograms of research cargo going up, about 600 kilograms of research samples coming down, supporting about 280 investigations active on board the International Space Station. So the question is why? Why would we do all of these things on the International Space Station? And a key for many of these different investigations is what we call microgravity. Going into space, being in that permanent free fall, so it's as if there is no gravity there at all. In the physical sciences, that means that we can simplify the equations that we do with physics. Basically, we don't have to model convection. We don't have to model buoyancy. We can study processes there that we can't study that well anywhere else down here on Earth. Then in the life sciences, it's even almost more interesting because as you go to that space environment, you're leaving behind one of the strongest factors in the evolution of all life back here on Earth. And what's really interesting is not only do we care about those impacts on humans, on cells, on organisms, for what it means for future exploration of the human race beyond Earth orbit, but also because a lot of those impacts on humans look a lot like aging here on Earth. And so we can actually make advances that help make our lives better and healthier back here. Thanks.
And to pick up where Julie left off, I'm Mike Roberts. I work for CASIS. CASIS is the Center for the Advancement of Science and Space. We have the honor of working with NASA to support and manage the ISS National Lab. So in addition to the great science that's going up to the International Space Station to help us explore deeper into space, there's also science and technology projects that are going up there that have direct Earth benefit. And the ISS National Lab is that destination for those types of experiments. So we have over 20 payloads encompassing 40 experiments that range from the life sciences that Julie mentioned all the way through the physical sciences. And we're excited to have the opportunity to see these missions go up to space and translate back into Earth benefit. You'll hear more from some investigators coming up, but one of the projects I'd like to mention briefly that Julie mentioned, the effects of spaceflight everyone's familiar with. In addition to having great fun with food and water in that environment, you get to play with M&Ms and squirt water around. One of the things that happens is the physiological responses on the body. And those types of changes are of interest because they mimic some of the effects that happen here on Earth with some diseases and as we age. So you're going to hear about a couple of experiments later that are directly utilizing that microgravity influence that Julie mentioned to further their own research and improve understanding of human disease here on Earth.
Thanks. If we have questions from the audience, please raise your hand and we'll take them. Julie, I have a quick question for you. So I know you can't pick favorites, but do you have there's a particular experiment going up, launching tomorrow morning that you find particularly interesting to talk about?
Yeah, I definitely don't pick favorites, but there is one that's super interesting to me. It's called mouse epigenetics. It's an experiment flown by our Japanese space agency colleagues, and they're interested in looking at the effect of the space environment and radiation on epigenetics. So if you think of genetics as the words that code for how our bodies work, epigenetics is the punctuation, all of those extra things. And they're looking for the effects of the space space environment, including radiation and also microgravity, on those epigenetic pathways. Those mice will return home live, and that will be the first time we've returned live mice from the space station. And then they'll actually have the opportunity to breed back here on Earth, and that will teach us a lot about how space affects reproduction.
Michael, same question.
Again, difficult to play favorites, but one of the experiments that you're going to hear more about from the investigator coming up is an investigation called heart cells. So one of the neat things about the space environment is that affects biology, even the smallest biology. So even individual cells as small as bacteria, even things that aren't quite living like viruses can respond to that environment. So we know that on the human side, our immune system is affected by exposure to that environment. And in the case, there have been very exciting findings focused on the use of stem cells. Stem cells, as I think everyone's aware, are cells that have the ability to form any other type of cell. They have the ability to be pluripotent. And there's a lot of interest in utilizing stem cells for repairing damage that happens to us as we age. So there are investigations that are now going on to explore how microgravity can influence our understanding of the way stem cells proliferate, the way they grow in that environment, and the way that we can direct their differentiation to form different cell types for cell repair. Very exciting stuff.
Take a question from the audience.
Hi, good morning, or good afternoon. I'm Bill Carton, social media representing the Facebook SpaceX group. And my question for you is, what is the backlog of science experiments wishing to be flown? Is it a negotiation all the time to fill an allotted space, or is there a priority involved in terms of science versus consumables?
Yeah, so we do have a backlog. The primary limiting factor right now is crew time. So there is a lot of science that's funded on Earth that's ready to go that needs to wait until there's enough time for the crew to do those limited things that the crew can only do. Now, if an experiment can be done in an automated way, it absolutely is, and those experiments are ticking away all the time. That queue on the ground is prioritized two ways. First, by Congress. Congress has told us that we should use 50% of our resources for NASA's exploration mission and 50% for the national lab mission of benefiting life here on Earth. And so we set the priorities between those two buckets, and that influences kind of place in the queue. But we also work really hard across our partnership and to do creative things so that we can maximize the research on the space station and keep the queue as short as we can.
Quick follow-up. Then the commercial crew ability to get 7 staff on station that improves science availability will be fantastic.
Exactly. You said it better than I could have. Having commercial crew and going to a crew of 7 instead of a crew of 6 makes an enormous difference, because that one extra crew member will pretty much be doing research full-time, 24/7. Well, not 24/7, they'll sleep. But they'll pretty much be doing their research full-time, and also because that really streamlines all of the things we can do. And so it will make a huge difference. What will happen then is we'll, of course, start being limited in something else, but that's the goal, is to keep every single resource, whether it's crew time or up mass or facility space or data bandwidth, we want to keep all of that maxed out.
Appreciate it.
Thanks.
And the Dragon doesn't just launch cargo to space station, also returns it here back to Earth. Are there any science experiments coming back down that you particularly find interesting?
Well, one of the things, we have certain experiments that we round trip altogether so that they're on a very tight schedule. Also, we empty out our freezers of almost all the other samples. So one of the major things in the 600 kilograms coming home when this SpaceX returns will be all the blood, urine, and other samples from the crew members that are helping us understand comprehensively all the different effects of space on the human body.
Hi, I'm curious, are there any CubeSat missions going up on CRS-9 to the ISS on this mission?
I have to think. I'll have to get back with you on that. Right now I'm blanking. With 280 investigations, I know at some point, and then sometimes I forget, so it's slipping my mind right now. Sorry.
Thank you.
Stump the project scientist today. Okay, any other questions? Julie's going to be sticking around. If there's any other questions come up that pertain to her, we can certainly answer them. Thank you, Julie. Thank you, Michael. Appreciate it. Okay, if you're watching this at home, you have questions. It's not just for those here in the room. You can please use the hashtag AskNASA. If you're on social media, you can use the Twitter or other types of social media platforms, Ask NASA. Our next speaker is Sarah Wallace, who is a NASA microbiologist. Sarah.
Thanks. I'm really excited to get to talk to you today about the biomolecule sequencer payload, and I'm really excited to show you this DNA sequencer. But where? Oh, wait, I forgot. It's right here. So this is this exceptionally small device, has massive research potential. So this is the MinION by Oxford Nanopore Technologies, and it will be the first ever DNA sequencer in space. I'm going to just sit it here for the rest of that. So while it sequences fundamentally differently than bigger sequencers that I couldn't carry around in my pocket, the end result is the same, and that's a sequence of the nucleic acid bases A, G, T, and C. And as you can see, its small size is ideal for spaceflight, and also it doesn't require much power. We're just using a tablet that's already on the ISS to power it via USB connection. So perfect for spaceflight. So it can sequence, we called it the biomolecule sequencer because we didn't want to limit it to just DNA. So it can also sequence other biomolecules like RNA and even proteins. But DNA is a really important thing to be able to do to sequence it, and it gives us a lot of capabilities. For example, identifying microorganisms that are in the environment, diagnosing infectious disease, monitoring crew health, and possibly this device could one day help us detect nucleic acid-based life elsewhere in the universe. So this could really be huge in exploration of other planets. So as I mentioned, it could also do RNA. So we're not doing that right now, but someday we hope to use it. And so if we could sequence RNA, that gives us information back on living systems and to how they're responding to the environment. So turning on and off different genes and how to best to live in that environment. So the ability to do that would give us a ton of research potential to understand how things are responding to microgravity. So my colleagues and I at the Johnson Space Center, Aaron Burton and Kristen John and Sarah Stahl, along with the rest of our science team, astronaut Kate Rubens has been very involved with it. And then we have members at the Goddard Space Flight Center and the Ames Research Center, and even in academia at Cornell and UCSF. We've designed an experiment in which astronaut Kate Rubens is going to sequence the DNA from a virus, a bacteria, and a mouse. And these are all going to be contained within one sample. And while she's sequencing it on the ISS, we're going to be doing the exact same thing in our lab at the Johnson Space Center. And then our goal is to compare how well the sequencing is working on space with how well it's working here on Earth. And so we're really excited to get the sequencer working. And I just should mention that it was this device, because it's so portable, it was really important in the Ebola outbreak. It could be deployed in the field, and people could be using it to detect Ebola in blood samples within hours. And we hope that this technology could do the same thing in space. We currently don't have the ability to do that. So if an astronaut gets sick and it's an infectious disease, it's between them and their flight doc to figure out what it is. So huge potential with this. And we're also developing the sample prep process so that the crew could use it to go all the way to collect a swab or a sample and do everything they need to do to be able to sequence it. And that process will be going to NEMO, NASA's underwater extreme habitat off the coast of Key Largo on the ocean floor. It'll be splashing down tomorrow as well. So very exciting day for the sequencer tomorrow. And we also have a team member taking it to the Arctic to look at the possibility of in another streaming virus environment. So a lot of good stuff coming up. So stay tuned as the first DNA sequences from space are sent back to Earth in the coming months.
Sarah, quick question. So you mentioned that if somehow this could be used to detect life or elsewhere in the universe, how exactly can the DNA sequencer do that?
So this DNA sequencer functions vastly different, as I mentioned, than any other sequencer. So really what it's detecting is you have an ionic current that's created while you're sequencing. And all that it detects is when a nucleotide passes through the nanopore, and those are biological proteins, an actual pore that the DNA is passing through. So you have a current, and then as something's passing through, it changes the current. So it's reading that change in current. So really, if you think about it, this could detect any molecule that would be small enough to fit through that pore and change the current. So my colleague that I mentioned, Dr. Burton, he's an astrobiologist, and he's wanting to look at, and his team, modified bases. So the bases that make up life as we know it are the A, G, T's, and C's. So what if it was an A, but it looked a little bit different than ours do? This device could potentially detect that, and that's the kind of research they're working on right now.
That's awesome. Do you know if they're looking at potentially they're testing it first at space station, and if it works there, then they would think about maybe putting on a future mission elsewhere?
Right. As right now is the time to utilize ISS for both crew health, which is what I'm interested in, and possibly going far beyond ISS one day, which is what he's interested in.
Right. Any questions for Sarah in the room?
Hey, Sarah. Over here?
Yeah.
Thanks. That device looks really awesome. Any chance that you can show us pictures of it on the screen?
Yeah, there should be another picture. There it is. So there it is, opened up. And that flow cell that's right inside, that's where all the sequencing chemistry takes place. And so it just pops in and out. And each one of these is good for about 48 hours. And you can really put as many samples as you want on it. You just flush it out in between. And as soon as you start getting your data in real time. So as soon as you're happy with your data, you can flush it out and put a new sample on.
My question was, how long have you been working on this project?
Well, this was actually part of Johnson Space Center's Class 1E. And what that means is normally all flight hardware is Class 1, and that's a very strict regimented process. But things that are more experimental in nature, like this, JSC just rolled out this 1E process, which is a quick way to get science to the ISS. So we were kind of the guinea pigs in this. We were given authority to proceed last February 2015, and we handed over our hardware last December. So we did this in 10 months.
Wow.
Yeah.
It's amazing.
DNA sequencing has gotten a lot cheaper over the past 10 years and even just in the past handful of years. How much does this device cost to produce? And you mentioned the possibility of it being used in something like an Ebola outbreak. So in a setting in developing countries, how much could this cost? Could it be commercially available someday, I guess, is what I'm curious about.
Yeah, so I should have mentioned this is commercially available. So we didn't do anything to it. It's commercially available off the shelf, COTS hardware, as we say. So we didn't modify at all. The company, Oxford Nanopore Technologies, is obviously, the portability is what this is about, right? So they want to make it very, very easy to obtain. So the sequencer itself, you sign up with the company and they give you, and then each flow cell is $1,000. So it's kind of that $1,000 genome that we're at. So it's really, in the terms of what you get for it, it's a pretty cost-effective way for NASA to really be able, and based on the size and power and all that, really be able to put this technology on the ISS.
Hi, Sarah Marquardt. You had mentioned about that you'll be using that to test mice both on the ISS and both back at Earth. I'm curious what, if any, you have anticipated outcomes for that.
So the samples that we'll be sequencing on the ISS are ones that we prepared in our lab. And so we split them, and then so we'll have the identical one. The things we're looking at is just how well is the sequence look the same? Is the error rates the same? How fast are those sequences passing through the molecules? So we don't really expect any differences between the actual samples because we've controlled for that since this is the first time that this has been done. But what we're looking at is how well all the, ins and outs of the sequencer and how well it operates, and then how easy is it for the crew to operate? Is this something that's simple or something that we need to make more simple? So a lot of things we'll be looking at, but we expect the sequences to the DNA samples are the same. So
thank you.
I have a quick question.
Yeah.
So it seems like a fun coincidence or not that Kate Rubins is the astronaut on space station right now, and you're sending this DNA sequencer up?
I would say fun coincidence. We may have intentionally manifested to SpaceX 9, so we would line up with Kate. Kate's been on the science team from the beginning, and we have been so fortunate to have her because her passion is really in the science, and so from day one, when she came and sat down at the table with us, as soon as we just treated her like our scientific colleague, everything, it was great. She's been great to work with, and she's been really influential in those procedures that we're developing for spaceflight. So not totally coincidental that we're on SpaceX 9, but it really was fate that all this, we got the authority to do this last year, and it all lined up with Kate going to the ISS. So we're very fortunate for that.
Oh, my question is not technically scientific, but what advice would you give to a girl who would want to follow in your footsteps and work in space?
Yep, Thank you for asking that. I love that. This is obviously my dream job, and it's what I'm passionate about, and I'm so excited every day that I get to go to work. I would just say I didn't really do anything special, except I got interested in science at a very young age, and I am not good at math, so I knew I was not going to be an engineer for NASA. So I focused on biology from day one. And so I think that if you can just find what it is that you're passionate about, I mean, who knew that JSC hired microbiologists? There's a whole lab of us there. So whatever science, math, engineering, whatever area you're interested in, find it, pursue it, follow it, love it, and just stick with it and keep working towards that goal.
Awesome. Thank you.
That's great. That inspired me. Any more questions in the room? Okay, thank you, Sarah. Appreciate it.
Thank you.
Our next speaker is Rubik Sheth. He is a program manager, and he works in something called the phase change heat exchanger, and he'll tell you all about that.
My name is Rubik Sheth. I'm the project manager for the phase change HX payload, and also a thermal systems engineer from the Johnson Space Center. So what we're flying is an experiment to understand thermal control, essentially. But this is thermal control around the moon. We're going back to the moon. We're going to go back out of LEO, low Earth orbit, and we're going to try to do things that we haven't done in a very long time. So one thing that you have to account for, though, whenever you fly a spacecraft is thermal control. Robotic, manned, unmanned, a CubeSat, anything that you need to fly, anything that you use, the cell phones that you're using today, they require thermal control. So the payload that we're looking to fly is to understand how we could regulate temperature and make sure that our spacecraft, specifically Orion, the multi-purpose crew vehicle that NASA is developing today, could regulate temperature as it's orbiting the moon. The moon provides a very harsh environment. It actually ranges from plus 300 degrees on the sun side down to nearly absolute zero on the cold side. It is a very harsh environment compared to even Mars or any other place that we've sent things to. So to do regulation of temperature while making sure our astronauts are comfortable in a space vehicle is of utmost importance. So the phase change HX payload was built to help understand how we could put a phase change material heat exchanger to help absorb that excess thermal energy whenever a vehicle is right in between the moon and the sun where it's very hot, and then reject that energy when it has gone to the dark side where it's very cold and re-solidify that phase change so that it could be used again when it orbits to the sun side of the moon. So it's a form of thermal capacitance that's provided to the vehicle. This has been done before on the lunar rover, where we placed literally a block of ice, kind of like what you would put in your freezer packs. They just put that on top of some electronics and hope that the electronics would stay cool. So they did that then, and then on Skylab, we actually had a pump fluid loop. We actually flew coolant through the phase change material heat exchanger, but it actually worked inconsistently. It actually had some problems, and so this is the first time that we're actually going to try to redo that type of heat exchanger, but make sure that we could actually use it for Orion. So if you look at the video that we're about to show here, the video, so PCMs is a substance with a high heat of fusion. The heat of fusion is what allows me to store energy within something that could change temperature. So our phase change material heat exchanger payload, it's a double mid-deck locker that will be located within the U.S. lab of the International Space Station. It's a mechanically pumped fluid loop. It has an upper portion that I like to call the kitchen drawer. You could actually take the entire phase change material heat exchanger out and put in a new phase change material heat exchanger. As we develop new heat exchangers, we could fly up the new ones. We could test them out. We could put them into the hardware to make sure that it works and it could properly freeze and thaw the PCM or the phase change material within the heat exchanger. If we all run into PCMs at home. If you put a soda can inside the freezer and you forget about it, what happens? It explodes, right? So what we're trying to do is use candle wax or something like candle wax. It's specifically called N-pentadecane wax. And we're trying to use that wax, put it inside an enclosure, and then as it expands and contracts, we were trying to make sure that that expansion contraction doesn't essentially doesn't create that Coke in a freezer situation in the spacecraft. So that's about all I had. I was looking to take any questions if you had any.
And not just those in the room, but if you're watching at home, please use the hashtag AskNASA. Let me ask you something real quick before we go to the audience.
Absolutely.
So you talk about the soda can in the freezer. It made me think, is this the work you're doing now, is that something also could be applicable to real-time applications here on Earth?
Absolutely. So one of the things that the company that we're working with, United Technologies, the other thing that they're looking at here on the side is trying to understand how they could put these PCM heat exchangers into HVAC systems. If you could help store that energy in an HVAC system, you could essentially bring down that requirement on how much energy you have to use to help cool your house or heat your house throughout the year.
Would a system like this, for the next step, be tested on a satellite or a CubeSat, or would this directly from the ISS be integrated directly into Orion from your findings?
They are actually going to integrate it directly into Orion, assuming that it works out and we prove that it actually works on orbit. But we're looking to, as quickly as possible, test this, and then by December make a decision to see if we can put it into Orion.
Thank you.
Mm-hmm.
Okay. Question in the front over here.
Hi, thanks. I'm an engineer. I use some phase change thermal grease materials. This looks like it's also great for home use for time shifting the demand to maybe a time where there's cheaper electricity, and definitely on Orion around the moon, the time where you have some capacity to get rid of it.
Right.
I'm interested in the material. What's the coefficient of expansion compared to water or soda? It's not the same expansion?
It's actually,
ratio.
so for N-pentadecane, it expands by about 9%.
Real? Oh, that's
worse.
worse.
It's actually pretty bad. But the interesting thing is that water expands when you freeze it. For wax, it expands when you thaw it. So we have almost like an inverse problem there, but it's still the same problem, right? It's expanding, and we're having to accommodate that expansion within an enclosed volume and make sure that our heat exchanger isn't catastrophically damaged.
But it's pumpable when liquid. It's not so viscous.
Absolutely. It's like mineral oil. It's almost the consistency of a mineral oil.
When frozen? Or
when
solid?
frozen, when solid, it's like candle wax.
Got it.
It's like candle wax,
Got it.
right? Yeah.
Thanks.
No problem.
Okay, thank you. Any additional questions in the room? And for those watching at home, remember you can use the hashtag AskNASA if you have a question, and we'll do our best to get to and answer it. Thanks, Rubik. Appreciate your time.
Thank you. Thank you.
I think we're going to a video next.
My name is Peter Lee, and when I'm doing research or looking at space, my other hat I wear is as a heart surgeon at Ohio State. This is my colleague, Arun Sharma, and he's finishing up his graduate studies at Stanford. We're really excited here to talk to you about our research where we're looking at heart cells up in space. The rationale for this is quite simple. As you all know, there are a lot of systems, pretty much every system in the body is affected by the microgravity environment, and the cardiovascular system is no exception. And in particular, we're interested in the heart. We know something about what happens to the heart, but unfortunately, all the studies have been limited to studies in humans or whole animals. And so we know very little about what happens at a tissue, cellular, or molecular level. And a lot of that was limited by what we can do in terms of doing cell studies in space until now. And so the rationale for this is look at what happens at that level so that we can understand mechanisms that will ultimately, hopefully, find ways to prevent or treat disorders or diseases that parallel what happens in space on Earth.
Right. So as our new model system to be able to better study cardiac function in space, we're actually utilizing a pretty revolutionary new piece of technology that's been developed over the last 10 years or so in the field of stem cell biology. And these are called induced pluripotent stem cells, or iPSCs. basically what we're able to do is to take a small sample of a person's own white blood cells and transform those cells back into a stem cell form, a pluripotent form, meaning it can turn into what other cell type you want, whether it's brain cells, liver cells, and since we're a cardiovascular research institute, we're interested in studying heart biology, so we can transform these iPSC stem cells into beating heart cells that you can actually see contract under a microscope or sometimes with your naked eye, which is really cool to see. I think if you want to show that video again, we can emphasize that point a little bit. Yeah, so they're a really fun cell type to work with. And the big advantage of these cells is for the first time, we can mass produce these heart cells without performing surgery on a patient, which is a very powerful thing. So what we'll be doing is sending these cells to the International Space Station. We've actually been collaborating with Bioserve Space Technologies, which is an engineering company from the University of Colorado, to develop a custom apparatus for actually growing these particular cells in a microgravity environment. And once these cells are actually on the International Space Station, they'll be up there for 1 month, and they'll be maintained by Dr. Kate Rubins, who is one of the most recent astronauts aboard the ISS. What she'll be doing is looking at these cells and seeing how they're going to change in terms of their beating rate, in terms of their cell shape, size, and also changes in gene expression. So we're really excited to be able to utilize the ISS because it provides us with a novel resource and a novel environment in microgravity that's not really available to us here on Earth.
Great. Any questions in the audience? Right up front here.
Hi. I'm curious. Obviously the pluripotent stem cells, it's so fantastic to not have to do open heart surgery to get those. I'm curious, though, is there any negative aspect to using pluripotent stem cells that you've turned into the heart cells as opposed to ones actually harvested from a heart?
Right, that's a great question. So there are some limitations associated with these particular cells. I would say the biggest one is that they're somewhat immature, meaning that they're not identical, they're not perfect replicates of the true adult human heart cells, which can take, years to grow and years to become an adult heart cell. And we're producing these within the matter of a few weeks. So that is probably the biggest limitation with these particular cells.
So would you say that the cells, because they're young, would they be comparable to a young person's cells, or because they're being produced from, you might have harvested like white blood cells from an older person, would they be that age?
Right, that's a great question. That's actually something that we're still trying to figure out right now in the field of stem cell biology, is that are these heart cells more of an embryonic-like state? And when you actually reprogram that person's white blood cells to a stem cell state, are you losing some of those age characteristics as well? That's something that we're still actively investigating.
Right. Thank you so much.
And to follow up on that, one of the reasons we have very little data on heart cells is because cells that are isolated from tissues, whether it's from a mouse, rat, or human biopsies, is that they're very difficult to culture, to grow, and to maintain. And that's why these iPSCs have revolutionized this field in just the last few years. And that's why we have this opportunity to do this for the first time, looking at heart cells in space.
I have a question. Does it matter who you collect the white blood cells from? Are you collecting them from a wide range of subjects? And are you growing them into heart cells on Earth and then transporting them? Or are they being grown on the space station?
Right. So we're actually collecting these from 3 separate individuals. So we'll be flying the heart cells of 3 separate individuals. They're actually produced in our laboratory at Stanford University. Okay. Mm-hmm.
Any other questions in the audience? If you're watching at home, please use the hashtag AskNASA. Okay, Arun, is this the first time we've done this sort of we've flown cells like this before on the space station?
Yes, I would say this is the first time that we've flown human heart cells, or cardiomyocytes, which is the fancy scientific term for the beating heart cells, in large part because, as Peter mentioned, we haven't really been able to maintain true adult heart tissue until this moment, until this time, until the iPSC technology that I talked about.
The actual experiment itself, when it goes up, is it just do you get the data from the Earth, down to you? Do astronauts do something with the experiment? What happens with it while it's on orbit?
Right. So while it's on orbit, Dr. Kate Rubens will be updating us on a weekly basis in terms of how these cells are behaving. Are they exhibiting any alterations in terms of their beat rate, in terms of their shape and size? So we will be getting somewhat of a live update from her.
Gotcha. Is that video that comes down, or is that a written report? I'm always curious about how the science on space station is actually investigated from the ground.
Right. It'll be a little bit of both, definitely.
Okay, great.
So we'll be getting some videos on separate occasions, and then also some of them will be fixed so that we can get the RNA and to study that when we get back, and then the rest will be brought back live for further analysis on the ground. And then the other thing, I mentioned this earlier, is the reason we're looking at these cells is so important to us is that we know what happens maybe at a heart level, but there's so many different factors that affect the whole organ when it's inside of a body. So when you're able to do it in a culture dish and isolate all these other factors, then you're able to just really nail down what's causing the changes. And so that's why we're really excited about this.
So with the results of this, could this kind of help us understand why there's so many skeletal changes and muscular changes with astronauts and just people living in the International Space Station?
Yeah, I think there probably is a fair amount of overlap. So we know a lot more about muscle and bone. And my background originally was skeletal muscle, and we looked at skeletal. But again, since we haven't been able to look at heart muscle at this level, this will be able to provide us some data that we can compare to these other types of cells. And I think we may be able to find treatments or things that are wrong that may provide some help in terms of treating these other diseases.
Thank you.
Go ahead in the back there.
Sorry. Is it just the United States working on this project, or are other countries or nations involved as well?
That's correct. Right now, it's just the U.S. These particular cells, this particular protocol to make these cells, is something that different labs around the world are also working on.
And that's just and this is, I do want to thank CASIS for the opportunity for us to fly this experiment. When the opportunity came up, they were looking for experiments that had to deal with stem cells. And me being in the cardiac field, and with Arun and Dr. Joe Wu, who's the PI, being in the heart field, it made sense to look at the heart and in space, and we happened to all be at Stanford, so that's how our team came together.
Awesome.
I had a question. What kind of applications, if any, would this have to congenital heart defect repair or prevention?
Sure. So what we don't know is what kind of changes exactly we'll see at a cellular molecular level. We know a lot of what may happen in certain disorders, and in the lab that Dr. Sharma and Arun and Dr. Wu has, they have a whole bank of cells from all different donors, and some of them have congenital disorders. And so what would be great is we can take these cells and especially look at the gene changes when we look at the RNA that's been fixed in space, and we can see if there's parallel with some of these disorders, whether they're congenital or acquired. And if we can see these parallels, maybe we can make that connection, and it may have some impact in terms of treating some of these in the future.
Very cool. Thank you.
Hey guys, Luke Fewer. So you were talking about sort of live updates. Is there any way, based on social NASA pages, that we can sort of follow this technology and sort of see what you guys see?
Well, I do have my own Twitter account, so I'll see if I can post some updates on a weekly basis.
Cool. Thank you.
That's right. So that's a.sharma, ADOTSHARMA. Follow me, please.
Please follow me. There's a question from social media over there once you get the mic. But in the meantime, I have a question, because you mentioned putting your team together at Stanford. How do you guys find out about these opportunities from CASIS? Does CASIS come to you proactively, or how did you learn about being able to do research on the International Space Station?
Well, I've always had an interest in space, always looking out for opportunities, and so I get updates as these opportunities come up, whether it's from NASA or CASIS. But I also know to actively look them up as well. So for those who have an active interest, you can go and look them up, whether it's on NASA CASIS or NSBRI, or that may be changing soon. So there are a lot of opportunities out there. You can actively look for them or get on the listserv, and you can get automatic updates.
That's great. Yeah, because there's limitless possibilities in science, and just knowing that opportunities out there is really important. Thank you.
The question is, what equipment are you using to monitor the cell behavior on the ISS?
So we are utilizing a light microscopy setup. So we'll actually be able to take videos, 15-second videos of these particular cells to monitor their changes in beat rate.
Okay. There's a question here on the right side of the room.
Hi, my question is, what is the complete lifespan of this experiment, and what would be the immediate follow-up?
Right, so the complete lifespan of this particular experiment, well, it's going to be on orbit for about 1 month, but we will be doing some downstream analysis for the subsequent 2 to 3 months to be able to compare our space-flown samples to actually a set of ground control samples to see those changes in gene expression that may actually have happened as a result of our exposure to microgravity, as well as these changes in beat rate, cell size, and cell shape.
Hi, Graham Grable from UGA Small Stat Lab. I'm curious, will these heart cells be flown back down to the ground to see how they react to being back on Earth with non-microgravity environments?
Yes, so there will be a sample of these cells that will be returned live, but they will be cultured for only a few additional weeks upon return groundside.
Last question. Maybe you kind of brushed on this already. How is this, in the end, maybe you didn't brush over, we went through it more thoroughly, but those at home, I mean, oftentimes people ask, like, why does the space station matter to me? How is this research really, do you expect to see results that are going to impact people with heart disease down here on Earth?
You know, sometimes there's no way to know exactly how it may impact a particular individual or a particular field, and that's the beauty of science. You know, we do a lot of science, some of which lead to discoveries that have a major impact on the world, and others may have smaller impacts. But you don't know until you do it. And the U.S. lab, the International Space Station, is a very unique opportunity. And as many people know, a lot of the changes you see physiologically is similar to accelerated aging. So instead of looking at cells and watching them age for months or years, we may be able to see a process that's similar to that in a very shortened span. And so the applications there for patients who may have heart disease or disorders, whether it's due to aging or some other acquired disorders, I think the potential is quite enormous. But in terms of how it'll impact an average individual, we won't know until we do the research. But it's well worth that because the potential return can be quite large.
Awesome. Thank you very much. Appreciate your time being here. Peter Bruin, thank you. Okay, let me bring up our next speaker, Bruce Hammer, who works on osteomics test flying to the International Space Station here on SpaceX.
Hello, everybody. Happy to be here. The project we're working on is looking at how bone cells operate in microgravity. And the name of the project is osteomics, and that's a combination of two names. Osteo means bone, has to do with bone, and omics has to do with genomics, proteomics, and metabolomics. So we're looking at genes, we're looking at proteins, and we're looking at metabolites of these cells. So when we came up with this project, this is based on, let me just get this straight. There's a video I'm going to show you about what we're doing, how we put these cells together. There it is. Okay. So part of the experiment is to look at sending up the cells in a frozen state. And the reason we do that is when cells are launched on a vehicle, they experience high G-forces and a lot of vibration. And what we're doing is sending up the cells frozen by very standard techniques of adding some cryoprotectant to the cell system. And in that video, you could see this is colleague Louis Kidder who was doing that. We then put the cells in a controlled rate freezer where we're reducing the temperature to minus 80 degrees Celsius. And when they're in this frozen state, they're essentially in suspended animation. So we then can then send up these cells and not have any kind of obfuscations from being exposed to high G forces and vibration. So when they do get to orbit, we've developed a technique with Bioserve Space Sciences where we can thaw these cells on orbit. And it's very standard practice where you take cells and initially get them frozen and then thaw them out. But in space, it's a very difficult process because on Earth, you can take a cell vial, put it in warm water, shake it around, and they're thawed out, and they're good to go to culture. When you're in space, you can't have an open source of water because a lack of gravity, the water is just going to fly all over wherever it may be. So what we've developed with Bioserve Space Sciences is a system where we would take cells, put them in a chamber where we have a piece of plastic on one side and a membrane on the other side, and the fluids are contained within. And that's what you saw Louis load up with media and cells. And when it's up in space, you can pretty much handle it and not have any problems with the media escaping from the system. So we are going to be growing these cells for approximately 30 days under different conditions where we look at mineralization and non-mineralization. And I should step back here and tell you what happens with bone. Bone isn't a static system. Bone has three types of major cell groups in there. You have osteoblasts, which produce bone matrix, osteoclasts, which eat bone, and osteocytes, which helps coordinate the osteoblasts and osteoclasts in communication with one another. So when you're on Earth or in orbit, you find that cells, bone is remodeled routinely. About 3 to 4% of your bone mass every year is broken down and rebuilt under normal conditions. When you're in space, there's a process known as demineralization that can occur, and there's a dysfunction between communication between the osteoblasts, osteoclasts, and osteocytes. And what we find is that astronauts have been losing bone mass at a rate it could be 1 to 3% a month, which is about what someone who has osteoporosis would lose in a year. So being up in space is a fantastic environment to accelerate this process of bone loss. And so if you're looking at what genes are being affected or what proteins are being affected or what metabolites are being affected, this gives us a fantastic environment to understand bone physiology. So that's one part of the experiment, is to do the study on orbit. But we're also developed on Earth a system where we could simulate microgravity. And if you roll a video, you could see a picture of inside of our magnet. You could see there's a spider sitting on a plastic nut floating in space, and there's an ant crawling around on there. And then the ant will jump off the nut, and you can see he's just floating around. So we can actually create simulated microgravity on Earth. And this is a video showing a grasshopper. And the reason we have that plastic nut there, he would stick to the walls of the magnet because he'd be freaked out by being in microgravity. But you could see how it looks just like if you put him up in the space station, that's how the behavior would be. So by creating this environment on Earth, we're now going to compare the genomics, the proteomics, and the metabolomics with what we see up at the space station. And hopefully, by having this ground-based model, we could use that to actually vet out experiments in the future. So if you have 10 experiments you need to run, and there's very limited crew time on the space station, you can run these experiments on Earth, see which has the most promise of payoff, and then select that to go up to real on-orbit science. So I'm open to any questions. If
it's you know, when you work at NASA, you come into work a day, you don't know you're going to actually see a grasshopper floating on a nut. You can't predict that when you walk in, but it can happen any day. I'm curious, before we go to the audience here, I know there's some questions. How does this, I know, we're going to Mars. NASA's working really hard to get us there. Is the research you're doing going to help in that endeavor with bone loss research?
Say it again?
For long-term spaceflight, for astronauts going to Mars or beyond, is your research going to contribute to that?
Absolutely, because what we're going to be identifying here are specific genes and proteins that are affected by being in a microgravity environment. By knowing that, you can then create compounds to actually take care of these abnormalities that are happening.
Okay, thank you.
Hi, I'm Kim Button. So the reason that the astronauts lose 1 to 3% per month, is that an increase in osteoclasts, or do you know why? That's
a good question.
Okay.
Excellent question. That's what we're hoping to answer. And the thing is, osteoclasts are more activated in microgravity. There's been some studies in the past that have shown that. And osteoblasts actually do decrease their activity a little bit. So you have this balance that's no longer there. When osteoclasts eat bone, it's normally a 2 to 4 week process where it would chew away at the bone, whereas osteoblasts need 4 to 6 months to replace the bone. So if you get any kind of break in the equilibrium between these two processes, you do have this problem occurring of demineralization of the bone.
Thank you.
Is this research you're doing, is it applicable to things down here, to osteoporosis? Millions of Americans suffer from osteoporosis. Is it going to help potentially cure or mitigate some of the effects?
Absolutely. What this will do, well, first of all, the numbers you're throwing that I've seen is $15 to $20 billion a year are spent on just people having bone fractures. And what you're looking at is roughly 40% of postmenopausal women will have a bone fracture, where about 13% of postmenopausal men, of older individuals can have significant problems and can get bone fracture. And so by identifying which genes are being affected by in this process, and also what proteins, we can then come up with a rational drug design that could attack these problems.
Okay, thank you. Any other questions from the audience? A couple there.
Quick question. Is this experiment a Earth-based experiment that could help astronauts, or is it an astronaut-based experiment that could help us here on Earth?
It's both. This study was funded by the National Institutes of Health back about 5 years ago is when we got this grant. And the focus of NIH isn't really astronaut health. It's health of individuals on Earth. But this has significant impact for both Earth-based and also astronaut-based individuals. And NASA is doing a good job in remediating some of the issues of bone loss. They do have astronauts now doing exercise on the space station where you're doing some impact exercise, like a treadmill where they have springs holding people down and they're running on the treadmill and taking. And I know of one astronaut, I don't know if it's as generally done, but taking anti-resorptive agents, which would actually shut down the osteoclasts, and the bone loss has been reduced significantly. So I think that answers your question.
Yes, thank you.
I think, hi, how are you? I'm Christine Fox. I feel like this is a follow-up maybe to his question as well. But when you mentioned that they were doing that type of, basically it's like a cardio exercise with a little bit, has any research done with them actually doing physical strength training against resistance for bone loss?
I'm not an expert on the physiology. Maybe Julie could maybe better at answering that.
Yeah, so astronauts do both running on a treadmill as well as high-intensity resistive exercise. And about a year and a half ago, scientists had published in the Journal of Bone and Mineral Research that if astronauts did the right mix of high-intensity resistive exercise, those are basically like squats and things like that on our ARED, our Advanced Resistive Exercise Device, plus had significant vitamin D supplementation, plus ate a lot more calories than typically they would do on their own. Then we did have astronauts coming home that had not lost bone mass density. Now their bone is still remodeling, and the structure of the bone may still have some risk for fracture. And so that's the thing that we're working on next at NASA. So really what we're seeing is this beautiful feedback between the bone loss in astronauts who are otherwise healthy, active, fit, no other problems going on, helping us understand bone loss. And then people here on Earth where we've got millions and millions of women struggling with bone loss, and we can use the information back and forth. When we make an advance on Earth, of course, we test it with astronauts, like an anti-resorptive medication, but also the things we learn from astronauts help us think differently about bone loss here on Earth.
Thank you, Julie. If you may or may not know, but have they ever done it without using cardio, just strictly high-intensity strength training?
No, we don't do that because that would be a significant risk to the astronauts. So they need to maintain their cardio fitness because they need that especially for doing the extravehicular activities, or the EVAs or spacewalks. So we need to keep their heart strong. If we didn't have them doing cardiovascular exercises of varying types, that would be a significant risk to their health. And of course, when they're doing the high intensity resistive exercise, they're also doing cardiovascular training. So it's combined.
Intensity is cardiovascular. That's
Right.
why I didn't know if they did just that. And
Yeah, so there's no way to really separate those two.
Gotcha. Thank you. Thank you.
Hey, Graham Grable from the University of Georgia Small Sat Lab. I'm curious on what you're thinking this experiment will show. Will it show that magnetic levitation is accurate enough to portray microgravity? What are your thoughts on the experiment?
That's a good. We won't know actually until we get the experiment back. However, we have done some preliminary work with the magnetic levitation, and what we're seeing is some pathways that aren't necessarily osteogenic related, it has to do with cell cycle, are being affected by the levitation. And so, again, we'll have to see what comes back when we compare the genomics on orbit to what's on Earth. And that won't happen probably. We're not going to get our samples back until January. It's only a month-long experiment, but because the cells are going up frozen, they can just sit up on the space station in a -80 freezer until the astronauts until the crew time is available to actually run the experiment. And so we're going to have to sort of wait and see. And we won't really have things analyzed probably until probably about May of next year.
Right, thank you. And as a quick follow-up, would there be any alternatives to magnetic levitation that you're aware of?
There is another. There are several ways of having cells experience, I wouldn't say a lack of gravity, but they have a rotating system where you could put cells on beads, then you rotate it around. And because the beads are falling all the time and rotating, you're averaging out where gravity is. It's sort of averaging out to zero. The problem with that technique is that you get a lot of flow shear around the cells, and that actually can have a bigger effect than microgravity would have on the cells. So we think the magnetic levitation might be a very unique and nice way of doing things because you don't have flow shear. Things just sit in the magnetic field and it could be sitting there for days or weeks or months.
Great, thank you.
Bruce, thank you very much for joining us.
Okay.
All right, our next two presenters are going to talk about the International Docking Adapter. We have Sean Kelly and David Clement. Sean, David, thank you for joining us.
Good afternoon, everyone.
Oh, hold on one second. There's a video playing. There we go.
Okay, very good. So it's not possible to talk about the International Docking Adapter without first talking about the International Docking System Standard. So this is the first implementation of the International Docking System Standard. I don't have to tell any of you about the power of the Internet. So one of the things that we first did as a goal was make sure that this was a truly publicly available international standard. So you can go to internationaldockingstandard.com and actually download this standard. And the whole purpose of it was to create opportunities for other entities, non-government entities, other governments that haven't traditionally been part of the space program to actually get their leg in the door on that. We're now working on other standards, and IDA is the first one that we've actually implemented, and it's going to be flying on SpaceX 9. I think we have a short video of that. So this is the International Docking Adapter in the SSPF, Space Station Processing Facility. This is in a orientation in our wait and see G fixture to make sure that to allow the commercial crew participants to come in and test against it. We do have rendezvous sensors on here. They worry about optical characteristics of it, reflections and things like that. So they brought in their sensor suites to test against the International Docking Adapter. This was part of the testing session. And I think we have some still images also of that we can call up now later tonight. And next image. And this is a view of it installed on the flight support equipment within the trunk. The ladder will not be flying with it. And I think at this point I'll turn it over to Dave. I think he's going to go through the installation sequence.
Well, thank you, Sean. Appreciate it. Boy, after listening to all the discussions earlier about all the science, as a guy who just provides hardware, it makes me feel, somewhat vindicated that I'm providing capability for something like this to be a permanent element to station. It's going to allow the commercial crew folks to dock in the future. So you guys get to do all the science. We like to provide capability, so it's gratifying to see that the capabilities we're providing is being utilized. So, as Sean said, International Docking System, it is a big darn deal. It's going to allow docking by commercial crew vehicles in the future without action by the ISS crew. Currently, right now, when we dock, we actually berth, and it requires the vehicle to come up to the ISS. It idles, and the robotic arm is then used to actually make the connection to the ISS. In this case, with the standard, we are now allow not only U.S. vehicles, but vehicles international. Anybody can build to the standard. It can be used on ISS. It can also be used for future spacecraft. So I think it's a real big deal to allow this. Docking is then accomplished by the host vehicle, which will come up and approach the IDA. It'll actually do a soft dock. So once again, it's the host vehicle that's in charge of its docking. A docking system is a lot more forgiving just by the nature of the mechanism. It allows for a lot of rate variations between the host vehicle, misalignments of the host vehicle. You'll come up and essentially do a soft mate, and then you'll be able to essentially align, wait for the vehicle's momentums to settle out. You'll then align the host vehicle to the IDA, and then you'll attenuate or you'll pull yourself in. When you get close enough, you'll be able to then set your latches and be mated. So that's really the function. And so to be able to provide this capability, I think it's a real big deal. So I'm happy to do it. I want to be able to show you, what Sean showed you was a lot of the interface and testing we've done. I have a video here that kind of shows how we're going to pull this and install this on orbit. As you can see, it's taken out of the trunk using the robotic arm, and it's manipulated. It's quite the manipulation to make sure, in order to get the IDA to the right port, you have to move that arm around the other elements on orbit. So you can see this. It's a lot of action that takes place here. We've orchestrated this many times. So for those who are going to make this occur, all the actions of the arm, all the controls of the arm have been worked out and orchestrated many times in simulations. And then as the IDA gets closer, it'll allow some time for idle to allow the reach thermal equilibrium with the PMA. And then as you can see, it gets close. You see quite the manipulations of the arm for this to bring this heavy device out, orientate this thing around and get us to the right port. And as it gets close, they'll bring it as close as they can, and then the EVA crew will actually come out and make the final installation. And once it's attached, the capability now exists, and it's ready to go. Any questions? Sean, anything else?
No, I think that covered it. As Dave said, we are utilizing the standard for exploration. So we've heard a lot of great science today, and a lot of great things are being done for science. But the space station is also an excellent platform for testing out new technologies and demonstrating hardware for exploration. The International Docking Adapter is one of those pieces of hardware that we'll be using, as well as the commercial crew docking adapters.
Thanks, Sean. Dave, I have a couple questions. If you have a question in the audience in the room here, please raise your hand high. And those at home, please use the hashtag AskNASA. My first question is, I always find it interesting that two spacecraft that have never met on Earth before can fit perfectly together in space. It's beautiful that engineers can do that. We talk about this docking standard. What organizations were involved in deciding what that standard was?
Yeah, so that's an excellent question. All the international partners that we have in the space station contributed to it. That's Canadian Space Agency, European Space Agency, Japanese Space Agency, and of course Russia and the U.S. all contributed to it. So it really was a huge collaborative effort to do that, to make sure that we were reflecting the not only the technologies that the rest of the world wants, but also the capabilities that they want built into the docking adapter.
Okay, thank you. Go to the room.
Afternoon. Thank you yesterday for the briefing over in the lab
about
the IDA. Because of ambient noise, I didn't catch everything you said, but if I remember right, you said the IDA 2 and 3 were better examples of the standard, and there were either nonconformances or some issues with one, which we all know was lost on CRS-7.
So, IDA 1, actually, after we went and we're building IDA 2 and did some of the interface testing that Sean was showing here, we were careful to go take another look at some of the features. Like you said, this thing has to fit together like a glove. There's very tight tolerances. So we went and looked at some of the strikers on the way that the interface between the docking system and the IDA may happen. Mm-hmm. And so we did make some slight adjustments to fine-tune those targets. Mm-hmm. IDA 2 and IDA 3, we dialed them in manufacturing and double-checked and triple-checked that they were right dialed in.
Excellent. So the standard was modified a tiny bit between instances, or was it within tolerance and you changed the target? So
in this particular case, we didn't change the standard. We dialed it in. But in some cases, during the development of the IDA and the IDSS, we did make changes to the standard as that whole evolution occurred. So some of those were during the program development. We saw there was a lot of fine radiuses along here, along the pedals, and so we did make changes to the standard as the program developed.
Yeah. Excellent. Thank you.
Good afternoon. My name is Colleen. I'm from Colorado Springs. I was curious, you're talking about this being an international docking unit that we can use and even in further exploration. Do you think this is something that maybe if there's a lunar base ever gets established, or when we do get to Mars, that this system will just be adapted, or do you think there might end up being something completely different?
Yeah, so the current agreement among all the current space station partners is that this will be the implementation that we use going to deep space, cislunar, or any other exploration activity. And it's because they've already invested in it. So when we started the standard, there was one docking system fielded in the world. That was it. That contained a lot of proprietary capabilities, things that that you couldn't go out and build without going back to the original manufacturer. So one of the major goals of this was to put it in the public sector and make sure that it was a standard that anyone could use and all the information was publicly available. So with that said, we don't know who will use it for exploration, but that's part of one of the enabling capabilities of the standard and putting it on the public sector. Some small company in Arizona, some small country somewhere in the world that today doesn't have anything to do with space may say, hey, I think I can do that. I think I can build that. So that's part of the enabling capability of putting international standards out there.
Awesome. Thank you.
Hey, Graham Grable from the University of Georgia's SmallSat Lab. I'm just curious on what type of testing model or testing procedure you guys have to go through to make sure that the IDA is ready for launch and installation onto the ISS.
Well, quite a bit is the short answer. We did a lot of interface testing on both sides of the interface because clearly we have to prepare not only the ISS side, which is here, and make sure that it's going to attach to ISS, but we also have to make sure that the docking system and the host vehicle will be able to attach to it as well. So we do interface testing on both sides. In addition to that, we also did a lot of testing just to make sure that it was going to go in SpaceX trunk all right. We had a lot of testing. We have components here, the stanchions that are just used to mount it to the FSE are here. They're not part of the actual operation on orbit. We also had to make sure that when we use the robotic arm, that the interface to the robotic arm to pull it out of the trunk was going to go work. So every aspect, every interface related to making sure that IDA was going to go on the first time was tested.
Thank you.
Okay, I see a couple more questions in the room. Before we get to those, I have a question for you. So you showed in the video that it was the robotic arm was going to dock, going to attach the IDA. Is it for autonomous spacecraft or ones that need to be docked using the robotic arm? Is there a difference, or is it's easier on one than the other? It's adapted for both?
Well, I'll let Sean, as far as the installation of the IDA, it's a combination of the arm and the crew that will install it to ISS. But from a host vehicle standpoint, no action by the crew is ever taken for a docking after this. I mean, the host vehicle comes up. It has the active part of the system. This is a passive ring. So it'll extend a Stewart platform, which is a 6 degree of freedom platform. It kind of scissors out. The two sets of pedals will kind of meet, and it'll actually hit targets on the IDA. It'll declare itself captured, and then, like I said, then the automatic control system starts to do the alignment, the attenuation, and bring the actual host vehicle into ISS, where it then commands the closure of the latches.
Is this considered a more reliable way to dock than previous docking mechanisms?
Yes, and there are alternative docking mechanisms. There's probe and cone, which the Russians use for Progress and Soyuz, but this is androgynous, and that's one strong advantage of this over probe and cone. It allows you to reconfigure the vehicle into configurations you hadn't pre-flight thought of. So obviously, two probes won't go together, two cones won't go together. So having an androgynous system allows you to do reconfiguration of the vehicles and gives you a lot more flexibility for exploration. And one of the things we heard earlier is about crew time, and crew time is probably the most precious resource that we have on the space station. And by going to docking, we eliminate a lot of crew activities associated with berthing. We free up a lot more crew time for science, a lot more crew time for doing some of the more important things other than the docking operation.
Awesome. Thank you. Go to the room.
Good afternoon. As you said, this standard has been released into the public sector, and any company can now create IDAs. Since Boeing is the first to do it, from my understanding, will they have to test it before it's allowed to be installed?
So we do have as part of the space station program, we do have a very rigorous testing program for any visiting vehicle. So any visiting vehicle that has an active mechanism on it will be tested against our standard, will be tested against the specification. You probably don't care about it. It's called SSP 5808. That's not in the public sector, but that's levied on the commercial crew vehicles. And it delineates all the testing that they have to do as part of that.
Okay, thank you.
I assure you, we verified against the standard.
Yes.
I'm Cyber Sandy from Texas, and is there a list of different countries or companies that are wanting to dock in the future? Will we have access to that as public?
As far as developing docking systems? Or?
Yeah, is there anything in the works? Because
right.
Y'all will obviously regulate everything.
You're definitely from Texas, y'all. So, y'all back at you. So, right now, the Europeans are developing their own docking system. It's called the International Berthing and Docking Mechanism. That will be both berthing and docking capable. So you'll be able to actually berth a docking mechanism. The Russians are modifying their APAS system so that it'll be fully compatible with the International Docking System Standard. SpaceX is developing their system. Boeing is developing for NASA the NASA docking system. So we have that. And the Chinese have also publicly stated that they're working on an international docking system compatible system also. So that's one of the powers that we see of this standard. By putting it out in the public sector, we've gone from having one docking system in the world that was operationally used to now there's 5 actively being developed. So that increases both competition, also increases flexibility and opportunity for commercial companies.
Congratulations, that's incredible. Thank you.
Let's go to the back of the room.
Hi, Anthony and Swasti. You guys showed us a few models yesterday at the Space Station Processing Facility. Could you elaborate on the role 3D printing played on the IDA development and design?
Sure, I'll be happy to. Like I said yesterday when the question was asked, we built that, made a decision to go build that full-size 3D printer mock-up. And it was quite a challenge at the time, but we used it to make use, to try to, as a model to help us in our manufacturing. A lot of times you'll release engineering and you have to look at it from a producibility standpoint. We use that 3D model to take out to our manufacturing folks and say, hey, here's how we're going to put this thing together. And it serves as a great purpose for looking for obvious interference, looking for better ways to put this thing together. And that's really what we used the 3D printer model for, was as a manufacturing aid. And it served well in that purpose.
Thanks.
Graham Grable again. Just to clarify, when you say that companies and different organizations have the standard open to them, do you mean that the IDA itself is open source, and so like maybe the public could make fun 3D prints like that for their home? Is that what you mean by that?
The interface standard. So the International Docking System Standard is an interface standard. So it's just how the interfaces go together. That was by design. It doesn't talk about the implementation behind the interface, whether you use electric motors or a cable drive system or something else. And the reason for that was because we didn't want to constrain the technologies. So if we put that in the standard, then in order to be compliant with the standard, you would have to implement that particular way. So we tried to strike a good balance between putting enough information in there so that somebody had maximum freedom to go off and build their implementation, but also provide enough details to make sure that when those interfaces came together, they would absolutely work. As far as the IDA itself, most of that is that intellectual property is owned by NASA, but not all of it. As part of some of the subcomponents, subassemblies, some of the existing vendors, such as Honeywell, have their own intellectual property that we've capitalized on for that. So the whole IDA itself is not publicly available.
Thank you.
But I like the idea of putting an IDA stereolift model out, and we can probably do something with our PAO colleagues to do something like that.
We have a question from social media.
What port is the IDA going to be attached to, or node on the ISS?
Node 2, IDA 2 will be attached to Node 2 Forward, and IDA 3 will be attached to Node 2 Zenith.
Do we know the timeline for when the attachment's going to happen after launch and everything? Is everything like that scheduled already?
There's some preliminary schedule for that. So the EVA crew is, assuming the launch happens on time, the EVA crew is planning on going out August 18th in order to do the final EVA installation. About a week or so ahead of that time, we'll start the robotics operations. Those are all done by the ground. Again, we're trying to make sure that we maximize the amount of crew time that's available. So the robotic extraction will be done by the ground controllers. They'll actually go and grab it, and then they move it a little bit, and it's a much slower operation than the crew doing it, but you're not time constrained. You send up commands, tell it to go to a certain the arm to go to a certain position. It goes there, you verify it, and then you go to the next position.
I have a last question. Could you hold up the model so we can see it on the camera? Flip it up so you can see the interior. How are astronauts going to fit through that little hole here?
Quite easily.
What is the diameter there?
So the tunnel is 800 millimeters width. You actually get about a little over 32 inches between the pedals.
Okay.
So the actual tunnel, you have to get through those pedals. So a fully suited astronaut can get through there. That's the intent.
Gotcha. Thank you. Not seeing any more questions. Sean, Dave, thank you very much for joining us. Really appreciate you being here.
Thank you.
Before we wrap up, I just have a couple programming notes. Course launch today or tomorrow morning rather, is 12:45 a.m. Eastern Time. You can watch that on NASA television at www.nasa.gov/NASA television. Of course, you can follow us on NASA on social media. A lot of great research was discussed here today, and I want to emphasize some of our social media accounts. If you're not already following those, CASIS is really important. They're talking about a lot of the private sector research being done on space station. We have, for Twitter, for example, we have ISS_research. That is talking about all of the research we're doing on our space station. And I think one of the themes we really learned here is a lot of the things we're working on is for the future of human spaceflight, but a lot of it is taking the stuff that's happening in space and how it relates to us down here on Earth. We're working off the Earth for the Earth. Thank you for all of our speakers. Thank you for our guests. Thank you for being here. And hopefully we launch 12:45 a.m. Thank you.